Hey there, pre-AP. Let's get started on some molecular genetics, shall we? Tonight we're going to talk about DNA history. That's just part 1 of this outline, 4.1. And then in a separate video, we're going to talk about the structure of DNA and how DNA replicates itself, DNA replication. Make sure you have a highlighter and a pen so you can take additional notes and highlight the really good stuff. So let's get started. So tonight we're going to talk about 4 very influential people or experiments that gave us some groundbreaking information about DNA. Up until the 1950s, no one really knew what DNA was. We'd never heard of the word DNA, deoxyribonucleic acid. No one knew of this. They only knew that somehow mamas and daddies, whenever they had children, their children had similarities to their parents. And as time went on and technology became more advanced, they decided, oh, maybe we can figure out how kids kind of look like their parents. Or sometimes they look like their grandparents a little bit. Brothers and sisters sometimes look alike. So these people were really curious about where do you get the fact that you look like your mom? Why do you have your dad's ears and your mom's eyebrows? Maybe you have your grandmother's nose and your granddaddy's toes. So this is very groundbreaking. Groundbreaking stuff. So go ahead, find Roman numeral 1. Please highlight Alfred Hershey and Martha Chase. Make sure you know the year, 1952. And this is a picture of them. This is Alfred Hershey, he's on the right. And then Martha Chase, she's on the left. Now this time in science, to be a woman in the science community was a big deal. Because still at this time, this is the 50s, it was not really acceptable for most women to be in science. So for Martha Chase to have the guts and just the gumption to say, hey, I'm a woman, I work in the science field, I'm just as smart as a man, and I can perform experiments just like they can, was really groundbreaking. And then a lot of people decided, hey, Martha Chase is pretty cool, she's gutsy, we like her. After that, other women, along with Martha Chase, decided, I'm a woman, and I would like to be a scientist too. So let's highlight some things on what was their experiment. So we look at letter A. Know that they worked with a bacteriophage. You don't have to know the T2 part. Just know bacteriophage. And all that is highlight. It's a fancy term for a virus that infects a bacteria. So actually bacteria can get a viral infection too, just like we can. And please know that this virus infected an E. coli bacteria. And you're thinking, E. coli? I've heard E. coli somewhere. Well, E. coli, if you've ever heard of food poisoning, sometimes it's from E. coli. E. coli actually is, pardon me, it's naturally found in the gut. It's found in your intestine, your large intestine. And whenever it ventures out of the large intestine via feces and ends up in food, and people ingest that food not knowing that there's E. coli from feces on their food that hasn't been cooked properly, then you get sick. Just a tad bit. Alright, let's look at big letter B. Please know that this became known as the Hershey Chase experiment. So think about Hershey bar, you're chasing a Hershey bar. So let's see what they did. So this is 1, 2, 3, 4, 5, and 6. Know that they used radioactive sulfur-35 to label the virus's protein outer capsule in one container. So they're explaining to you what they did. They had one container, and what they did is they labeled this virus's protein outer capsid. Outer capsid is just a fancy term for the outermost part of that virus. And they labeled it with a radioactive element. And those kind of tend to glow a little bit. because they're radioactive. So number 2, in the other jar, in the other container, they used radioactive phosphorus 32. And instead of labeling the protein, they labeled the DNA inside the virus. So that's very important. The first container, they labeled the virus's protein outer capsid. They labeled the protein. In the another container, they labeled the DNA. That's very important. So what happened in number 3? The radioactive viruses, please highlight this, they were exposed to bacteria. They introduced those glowing viruses to the bacteria. They wanted to know, hey, which one is the deciding factor here? What's going to actually infect the bacteria? Is it going to be the protein, or is it going to be the DNA? So after they did this, number 4, the radioactive sulfur container, that's the one with the protein, the radioactive sulfur did not enter the bacteria. It remained outside of the bacteria, saying that the virus did not infect the bacteria. So out beside that one, please, right beside number 4, please put virus did not infect bacteria. This is container number 1. But on number 5, the radioactive phosphorus container with the radioactive phosphorus did enter the bacteria. Why? Because remember, the other container that had phosphorus in it, that radioactive glowing phosphorus, they labeled the DNA, not the protein, the DNA. So highlight, when they reproduced that bacteria, the virus, whenever the virus reproduced inside the bacteria, it reproduced viruses. Because remember, whenever you reproduce, you have to copy your DNA. So that's what that virus did. It copied its own DNA. And what it did is that virus replicated and reproduced so many times that it overcrowded the inside of the bacteria. And that's why it came out of the dead bacteria. It just blew a hole in that bacteria and just exploded like a virus fountain. And highlight, the dead bacteria were radioactive from the phosphorus it possessed. Now, why do I have to know this, Miss Minor? Well, here's the point. Number 6. This proved that with 100% accuracy that DNA was the transforming agent, because it carries information from one generation to the next, not protein. For a long time, people thought protein was the carrying agent that carried your traits, characteristics about you from one generation to the next. Well, with Hershey and Chase, they came to the conclusion, without any shadow of a doubt, it's not protein, it's DNA. So here's a picture of that bacteriophage. Kind of looks like a little alien from outer space. And that glowing that you see is the radioactive phosphorus. So it actually does glow. And you can actually see, right in here, you can see the DNA being, you can see the DNA actually being injected into this bacterial cell right here. They're injecting their DNA. They keep their DNA up in here, inside their bacteriophage head. They call that a head. This is the tail. And all they do is they just deposit DNA in here. It infects the bacteria. The virus replicates and reproduces several times. And then this bacteria is actually going to explode, because it's going to die. Poor little bacteria. So this was probably what we would see today. They didn't have electron microscopes back in the 50s. I'm sure that Hershey and Chase would have thought, man, this picture's awesome. So this is just a picture of what they did. The first batch, nothing happened. Second batch, something happened. Alright, moving on. Erwin Chargaff. So what's so important about Erwin Shergoff? Well, he actually preceded Hershey and Chase. So please highlight his name. And this is what he discovered in 1947. This was right after World War II. This is whenever the United States went over to Europe, and whenever they had former Nazis that were scientists, and they didn't want to prosecute those people because they were innocent. They had nothing to do with the Holocaust or any type of war crimes. They were just being held hostage under Hitler's rule. They actually went over there and said, okay, we don't need to prosecute them. We just need, if you would, just kind of pardon them. They didn't do anything bad. Let us take them back to the United States. We'll make them change their name, possibly, and then they can work in our lab. I don't know if Erwin Shergoff was one of those people. Don't know. You'll have to look that up. But this is what he discovered. We need to highlight that he develops what became known as Shergoff's rule. And you are pretty fancy if you get a rule named after you or a law named after you, like Newton. So what is Shergoff's law? This law states, please highlight, that for all organisms, A equals T, C equals G. Now what? I remember talking about that whenever I was in 7th grade, Miss Minor. I don't remember. Go ahead, write this out there somewhere. A equals adenine. T equals thymine. Got that? Okay. C equals cytosine. And then G equals guanine. Very nice. So let's see what else we need to highlight. For example, because we're going to have a math question on this. Go ahead, write out beside number 1, put math question. For example, if a species has 32% thymine, you have equal amounts of adenine to thymine. So if you have 32% thymine, that also means you're going to have 32% adenine. You got to have an equal amount for that A to match up with that T. Now, let's see how much guanine and cytosine you would have. So if we look down there, that's where that 32 plus 32 equals 64. If you take 32% thymine plus 32% adenine, that equals 64% of your DNA is made up of adenine and thymine. So that also means that there's 36% that's unaccounted for. So how are we going to know that? We take 100% of your DNA minus the 64% that we found out that you're adding in your thymine, and you subtract that, and that's 36%. Okie dokie. Well, that 36% stands for both cytosine plus guanine. So if you want to know how much that is, you're going to take 36% divided by 2, and that's 18%. That's 18% cytosine and 18% guanine. We'll do another example tomorrow. Don't worry. Just be thinking. So there's kind of our Shergoff's rule. We're going to talk about uracil a little bit later. Don't get tripped up on uracil. Just don't let it enter your brain yet. Alright, to the lady of the hour, Rosalind Franklin. We're actually going to watch a video on her tomorrow called Photograph 51. It's a very important video that I always get my students to watch, and they always love watching it. So please highlight her name. She was in science, really big with DNA in the 1950s. We need to highlight what she did. She performed X-ray crystallography of DNA. She made pictures of DNA, and in the video they're going to talk about how she did that. So why do we talk about her? Because she did X-ray crystallography of DNA. She took pictures of DNA. A picture that she took was extremely important to helping James Watson and Francis Crick developed their model of DNA, which we're going to look at in just a second. So, why is this important? For number one, highlight the picture that she took. Why it's important. It indicated the double helix shape, that twisted ladder shape of DNA. And we need to know, number two, the part where it says, this picture also indicated that the nitrogen bases, those A's, T's, G's, and C's, point inward and are equal lengths in binding. Because it is always one pyrimidine, remember those are C and T, for one purine, A and G. And then, number three, the large areas around that X shape are the sugar phosphate backbone of DNA. You're probably sitting there going, huh? That's minor. What are you talking about? Do not worry. Just highlight it. It will all be explained tomorrow in the video. Do not panic. You'll be okay. I promise. I wouldn't lie to you. Alright, to our last person, then we're done. Ooh, this is Rosalind Franklin's picture of DNA. This X shape that you see actually indicates that there is a double helix. That twisted ladder shape. As you can see, my terrible picture of DNA. And what that meant is those little bitty kind of lines, that represented that those A's, T's, G's, and C's were facing inward. They were on the inside of the ladder, not the outside, like people previously thought. And all of this represents that there, and over in here, represents that all your sugars and phosphates, that deoxyribose sugar and those phosphates were on the outside. And it kind of explained the shape of DNA. Now this is very important whenever we talk about our last two fellas. So that's just another picture of DNA from the side. If we look in the middle right here, that's what she demonstrated. These were in the middle. These were on the side. Twisted ladder shape. Alright, to our last two fellas. James Watson and Francis Crick. We need to highlight their name, and in 1953, that's a very important date. Super important date. Why we talk about them is because they constructed the first accurate model of DNA. Before 1953. There were many that tried to make a model, many failed. So what they did, in big letter B, they used Chargaff's work and Franklin's work to fill in the gaps that they couldn't figure out. They were model makers. They made pictures. This is their model in the background over here. That's their model. This is Crick. This is Watson. He's English. He's American. So once they used both their works, Chargaff's and Franklin's, they said, okay, I'm going to take their work, and we're going to apply it to our work. And this is what they found out. Big letter C. The double helix backbone is composed of phosphorus and a 5-carbon sugar called deoxyribose. That supports the outside of the ladder. Excuse me. Then what they figured out in D is that the rungs of the ladder, the steps that you would step on to climb up or down the ladder, would be that purine base plus a pyrimidine base, that A and T, that G and C. And they found out that what holds those A's and T's and G's and C's together in big letter E is hydrogen bonds. This is what holds that A and T and G and C together. So, and then it twists into a double helix shape. And remember, please highlight that hydrogen bonds are weak. We need them to be weak because we want to be able to open up this DNA and to be able to copy it and make proteins from it. Alright, that's part 1. We're going to watch photograph 51 tomorrow. So I hope to see you tomorrow. And have a good night, guys.